All right, so we're going to continue talking about molecular genetics. In a previous lecture, we talked about DNA replication, which is very important for mitosis and meiosis, and during the S phase when we have to make copies of the DNA. And we learned some of the details about that. And so we're going to see that what we talk about today, you're going to have some very similar details and some similar phenomena. And so what we're going to talk about starting today, or in this lecture, is protein synthesis. And the first part we're going to talk about is transcription. Once we talk about transcription, we're going to talk about processing mRNA, then translation, and the universal genetic code. So again, I've broken this up into a couple of different lectures, just to make things a little bit tighter and easier to digest, and also there's just some logical spots to break this thing up. Now, why is this important? For the same reason we talked about last time. You know, we've unlocked the genetic code. We're learning so much about biology because we can understand genetics at the molecular level. And so for a 21st century biologist, this is all very important, that you have a good understanding of this process. And some of this may be a review for some of you, and that's awesome. But remember our mantra: DNA makes RNA, makes protein, which you see here. Now, DNA making RNA is called transcription, and that's the subject of this lecture. Then once you have a piece of RNA, that RNA gets translated into protein, and that'll be in our next lecture. And one of the points that we'll make is that, you know, when you make RNA from DNA, those are both nucleic acids, and so we call that transcription. And you know, transcribe is to copy down, is another word for that. Or you know, if you talk about the court transcripts, when they write down everything in court, it's just a record of things in the same language. And since these are both of the same type of molecule, one gets transcribed into the other. DNA gets transcribed into RNA. But you'll recall that proteins are a very different kind of molecule. They're not a nucleic acid, they're a polypeptide, they're made of amino acids, and it's a very different type of molecule. And so you're translating the RNA. And so when you translate something, like when you translate a language, you go from one language to another. And so that's kind of the metaphor here. You're going from the nucleic acid language of RNA to the polypeptide language of protein, so you need to translate it. So that'll be in a future lecture. So a gene is transcribed into RNA and then translated into a protein. And so remember that was our definition of what a gene is. It's a portion of the chromosome that gets turned into a protein. And so by definition, each gene gets transcribed into an RNA molecule, and then that RNA molecule gets translated into a polypeptide. Now, that polypeptide might have a biological function by itself, and so it becomes a functioning protein. Or that polypeptide might have to combine with other polypeptides to make a functioning protein. You remember quaternary structure, like hemoglobin, collagen, where you have to have many polypeptides that come together? So that's another possibility. It's also possible that it gets translated into a protein and that protein doesn't work. And so, for example, some recessive traits, like white flower color in certain flowers, you know, the gene gets transcribed, the RNA gets translated, you make the protein, but the protein doesn't function. And so it doesn't create the proper pigment, or it doesn't lay down the proper pigment, or something like that. So that's another possibility. Okay, so the first step in transcription is you got to find a gene. And you say, well, that seems pretty easy. Is most of your DNA genes? Actually, no. Most of your DNA doesn't code for anything. Now, all this non-coding DNA likely has other important, like structural functions. But the majority of your DNA does not get transcribed. Consequently, the genes are just kind of here and there and hidden. And so the first step is finding a gene, finding an area that needs to be transcribed. So how do you find a gene? Well, you look for a unique sequence called a promoter. And so a promoter is usually a few DNA base pairs, but they're a signal that this is the start of a gene. So a lot of times when you're sequencing DNA and you're trying to study an organism, you'll look for known promoters, and wherever you find them, then you figure that the sequences after that promoter is some kind of gene. And so then you can look and say, well, what is this gene and what does it do, and things like that. So the enzyme that's going to be important here is RNA polymerase. So you remember DNA polymerase was very important for building strands of DNA. RNA polymerase is what builds strands of RNA. And so this RNA polymerase finds a promoter, which tells it, hey, this is a gene, and so then it starts transcribing after that promoter. So how many genes do humans have? About 22,000. How much of your DNA is non-coding, is not genes? About 98%. So, like I said, that 98%'s got some structural function, but it is interesting that you've got these big long chromosomes and most of them are non-genetic. Okay, so when it's time to transcribe that gene, you can kind of break this down into 3 simple steps: initiation, elongation, and termination. So initiation is when RNA polymerase binds to the promoter and starts the transcription process. Elongation is just what it sounds like. You're elongating the RNA. And so the RNA polymerase builds the RNA bit by bit by reading one strand of the DNA. And then finally, there's usually a unique sequence at the end of the gene. And when the polymerase hits that sequence, that tells it to stop transcribing and stop building the RNA. And so here you can see a piece of DNA, and the RNA polymerase is bound to the promoter. That's initiation. The DNA is getting unwound and opening up, just like we did in DNA replication. When we replicated the DNA, we split those strands apart, and now we're doing the same thing. But whereas in replication, we worked on both strands because we needed to replicate both of those, in transcription, we're only working on one strand. The template strand is the strand that we're going to read to make RNA. The other strand is not used in transcription. And so elongation is when the RNA polymerase reads the template strand and builds an RNA. Now note that RNA is built just like DNA. It's in the 5' to 3' direction for the same reasons. You can only bring in a new nucleotide to the 3' end of the growing strand. And so if you look at the labeling on here, you can see which end is the 3' and which end is the 5' for both DNA strands and for the RNA. And you can see that it grows from 5' to 3'. Then finally you have termination. Whenever the polymerase reaches a unique code that tells it this is the end of the gene, it stops transcribing, lets go, and now you've got an RNA transcript. And so when you take genetics, you'll talk a lot more about these promoters. But there are some very common ones. This one's called a TATA box. And so you can see it's just a TA, TA, AAA. It's several TAs in a unique sequence. You see, it's not a very long sequence. I mean, it's longer than what they show here, but anywhere you find one of these promoters, you'll find a gene. And so here they're showing these things. These are other proteins called transcription factors. And so these bind to that promoter, and then they assist, they help the RNA polymerase to bind and start transcription. And so RNA polymerase just opens up the DNA, adds nucleotides to the 3' end, and grows that RNA based upon the template strand of the DNA. And that's what you can see here. And so now you can see how we go from DNA to RNA. This makes perfect sense, right? They're both made out of nucleotides. They're made out of 3 of the same nucleotides. RNA uses uracil, whereas DNA uses thymine, but they're very similar molecules. And it's the same rules as before. A's with T's, or A's with U's, and C's with G's. And so that's how you can read a strand of DNA and know what RNA to make from it. And RNA polymerase is what's doing this. And so if you look here, you see that when the DNA has a T, you can stick an A in the RNA. You can use adenine in RNA. G's and C's, just like before. But then if you look on the DNA strand, wherever it has an A, it sticks in a U in the RNA, a uracil. And so there are no thymines, there's no T's in this RNA. So you can see how it's still, though, the rules are still the same. Who matches with whom is still the same, and so that's how you can read this strand of DNA and make RNA. Pretty cool. And so this is, again, you can see it's unzipping the DNA and reading it base by base and bringing in those RNA nucleotides. Now remember, those RNA nucleotides, they're very similar to the DNA, they just have a different type of sugar. And you only need the one strand. So that's why we only have to read one strand of DNA, because RNA is only single-stranded. So once you've read one DNA strand, you've got everything you need. And like I said, the RNA polymerase reads the DNA and pairs those nucleotides with the DNA, and that's how it's able to build an RNA based upon the DNA. And that's really it. Now you know DNA to RNA. So a gene's found by looking for a promoter. RNA polymerase binds to the promoter, pulls apart the 2 DNA strands, reads one strand base by base to build the new RNA strand. And when it hits that stop sequence, then it stops transcribing the RNA, lets it go, and now you've got a piece of RNA. And it's reading the template strand from 3' to 5', but it's building the RNA from 5' to 3'. So here's a figure from your book showing the same thing. And again, you see the RNA is being built from 5' to 3'. Okay, so we built a piece of RNA. That's awesome. But in eukaryotes, we still have a little work to do. So at this point, it's called messenger RNA, or mRNA. You'll recall we talked about there's several different types of RNA that do different things. Ribosomal RNA is used to build ribosomes. We'll talk about transfer RNA in a little bit. Here we're talking about messenger RNA, because it carries the DNA's message. So all this happens in the nucleus, right? That's where the chromosomes are, so this has to take place in the nucleus. But you'll recall the ribosomes, although the ribosomes are built in the nucleolus of the nucleus, they go and do their work outside in the cytoplasm. They're either free ribosomes or they're bound to the endoplasmic reticulum. So the mRNA has to leave the nucleus to go find those ribosomes to go make the protein. But like I said, in eukaryotes, it has to be modified a little bit. So the first thing that happens is you put a little RNA on either end. You've got a 5' cap and a poly A tail. So the 5' cap is obviously on the 5' end. This is a short sequence. And then on the 3' end, you get a poly A tail, just a whole bunch of A's. So what's the point of all this? Probably to protect those ends. because you've got this long, straight molecule, it's vulnerable at the ends. That's where things are going to break apart, and nucleotides are going to break off. And so by putting these caps on there, those can break off, but the good part, the part you need of the RNA won't break off. Or those can sit there and protect the good part of the RNA from being, broken down. That's probably why these are there. And so that's what you're showing. They're just showing a little piece on the 5' and then the poly A tail is a little bit longer on the 3' end. Now the other thing that has to happen is that you have to get rid of introns. And so most RNA has these introns. These introns are little pieces of the RNA that are not coding, that also don't, they're not useful for building protein. So we got to get rid of them because we need to have a piece of RNA that we can use to build a protein. The other regions that actually are used to build a protein are called exons. So the exons are expressed. They're expressed into protein. That's good. That's what we want. Introns are intruders. They're in the middle of things, and we got to get rid of them. And so this is called RNA splicing. And so that's the next step that happens before the RNA can leave the nucleus. We got to cut out these introns and stitch together the exons. So we keep the good stuff and get rid of the intruders. And so that's what they're showing you here. They're showing you first, you got the 5' cap and the poly A tail. And then you've got these light pinkish regions that are labeled as introns. Although they were on the gene and they got turned into RNA, they actually aren't useful for coding for a polypeptide. So we got to get rid of them. And so that's what RNA splicing is. We splice out those introns. The remaining exons are all stuck together. And now you've got a functioning piece of RNA. Well, we could talk about this later, but an obvious question is, well, that doesn't make any sense. Why does your gene have, coding for these introns? And why do you waste time with these introns if we're going to just cut them out and throw them away? And a good explanation is, and a very likely explanation is because these are useful for creating variability, and they're useful for building different kinds of proteins. And so as these exons get stitched together, sometimes they get stitched together in different ways, or just by having these introns and exons, there's sometimes accidents are made and you stitch the exons together in different ways. And again, this is like earlier we talked about increasing genetic variability is good for natural selection. This creates more variability, and so your offspring might have a better chance of surviving in a changing environment. So it's probably got something to do with increasing genetic variability and giving you a better chance on an evolutionary time scale. At any rate, they're there, and we got to get rid of them. And so this is just repeating what I just said. The introns are removed and the exons are spliced together. Now the mRNA is ready and can leave the nucleus. So remember, exons are the good part. Exons are expressed. Introns are intruders, and they got to go. Okay, so that's mRNA processing. 5' cap, poly A tail, splice out the introns. The mRNA is now ready to be translated into a polypeptide, but that takes place outside the nucleus. So here's another figure from your book, just showing again the same thing that I just talked about. And so you can see a lot of the similarities here with DNA replication, right? DNA polymerase read the DNA strand, RNA polymerase reads the DNA strand. You read that to build a new strand, except this time you're just using different types of nucleotides, and you only have to read one strand. But they're all done in the 5' to 3', they all grow in the 5' to 3' direction. And so again, in genetics you'll get into a lot more details about this, but that's the most important idea right there. So In our next lecture, we're going to go ahead and take that piece of mRNA and translate it into a polypeptide. I can't wait. I'll see you then.